Today lesson is about the cell cycle. Okay, do you know what is a cell? So what is cell cycle then? When one cell is here, with the time pass, we can see two cells. So cell divides to make two cells. Now this event from here to here, it's called cell cycle. And these cells again have the capacity to further divide. So this goes as a cyclic fashion. This is called cell cycle. To make it easy, to learn it easy, we divide cell cycle into several stages. Precisely, we call these stages phases. So let's look what are the phases of cell cycle. So I try to draw a cycle, and here is the center. So this part is named as a G1 phase, and this one is again named S phase, and this one is G2 phase. After that, there is a phase here we call M phase. This one, M phase, is precisely the period cell divided into two. So when it starts from here, it goes all the way G1 phase, S phase, and then come back to G2 phase. So this G1 plus S phase plus G2 is called interphase. So interphase is quite long period of time. And this one, M phase, is a cell division. So we can say mitosis. So mitosis is the dividing of the nucleus, followed by the division of the cytoplasm. So that's called cytokinesis. Now these are the stages of the cell cycle: G1, S phase, G2, M, and cytokinesis. So when you look at this cell cycle, the blue color one, the round is, it's a quite long period of time. But M phase followed by cytokinesis is short period of time. In certain cases, some cells stop going for the next cell cycle. So we call them cell is enter into G0 phase. G0 phase is considered as cell is not dividing. Now briefly, we're going to look what's exactly happening in these phases. So what is G1 phase? It's a kind of a cell growth. If cell wants to make a two cell, the cell should increase in size. That means cell should increase its organelles. So then organelles start to divide first and cells start to grow. Now when it comes to here, next stage is the S phase. S phase is the DNA synthesis. So we learn this stage as a separate lesson under DNA replication. That's a semi-conservative way of DNA replication. It's completely different lesson. We learn it here. Once the DNA doubled, for example in human, the 23 chromosomes, pairs of chromosomes, become 46 pairs of chromosomes. In other words, 46 chromosomes become 92 chromosomes, then the S phase is completed. So S phase is crucial not to make any mistake in the DNA. Now S phase is completed. Next we enter G2 phase. Now G2 phase is again the phase the cell try to increase the growth and the organelles. And certain organelles, for example centrioles, start to double at G2 phase because centrioles play a big role in mitosis, cell division. So once G2 phase is completed, next coming to another important phase, mitosis. Again mitosis we are going to learn as a separate lesson. So this will be a separate lesson and this will be a separate lesson. After mitosis, then the cytokinesis, and now it's 2 cell. We can see 2 cells at the end. So it's done. This is the end. Now this is called the cell cycle. So 1 cell to 2 cell division, what's happening during this process is called cell cycle. So cell cycle we have divided into phases G1, S, G2, mitosis and cytokinesis. So during each phases, S phase will be a separate lesson and the mitosis will be another lesson. After this video, we need to look who is controlling this. As you know, if no one is controlled, cell divide without its control, this condition is called tumor or cancer. So there should be a precise and well controlled system are here, and we're going to learn that after this lesson. Thank you very much. If you like my videos, please subscribe.